Watch this. Watch this. Oh, boom, boom. I got it. I got 3.5 minutes of video.
Oh my god, that was the best thing ever.
Okay, watch this kid. I guarantee you he's gonna drill it. And down he goes.
You didn't even see that happen.
And that, ladies and gentlemen, is why we have snow days. So welcome to, I think this is the first live stream of the year, Mrs. Smith.
For this particular group of students, yeah. I mean, I've had live streams from class, but not with us
YouTube.
Together.
Yeah, not the YouTube live stream like we did during the shutdown of 2000 and I guess till it was still 2020. Not sure if he's watching it. In the past, when we did this in the spring, we would have had about 100 comments, but it's a little depressing.
I know, we're by ourselves. I don't know if students are going to pop in or if we're going to be teaching to ourselves. So,
no, that's okay. If we ourselves, that's fine. They can watch the repeat. I think it'll be beneficial for them to do that. If they do pop in, hopefully stick around because we plan making this a really beneficial thing for them. So the Killer King, long time thing. Who's that?
they always use these like names and we have no idea who they are.
I have an idea who it is, but
unless this is one of our previous students from last year that got a notification because they follow SOS and they were like, hey, we're coming back on to see Mrs. Smith and Mr. Suter. See, look, it is Mark Baker. It is last year. See, my students this year can't even get on there, but my students from last year are showing back up. I wish I could give you bonus points.
How about it? What do you think? Do we? I don't think we banter for too long. I think we jump to it because we like to keep these things short and neat and beneficial for the students. So, if you're watching the replay of this, we're going to try and make it easy for you to get through a couple of assignments. If you are coming live, we're going to make it easier for you to get through a couple of assignments. We thought we would do is take the plant and animal cell structure and the cell part and their jobs worksheets and just go real quick.
You keep repeating for me, so I really hope that you're not repeating, like echoing on the live stream, but I'll just ignore it and try to go.
You said it's difficult. You kind of cut in and out a little bit. I don't know. We never had a problem before. I don't know what that could be. I'll move that speaker back. Still the same, probably.
Are you going first? Do you want to go over the cell worksheet first?
Let me do that. We'll work out the mic problem, but we're gonna go. So I'm going to share my document camera.
I'll mute and you can.
Oh no, we don't want to share that. We want to get. How do I? It's been so long since we've done this.
Bye, Mark. Thanks for coming.
All right. So just really quick. We have a plant and animal cell. This is one of the first things that you have to do in the Senate once you get through the prokaryotic stuff. If you are still working in the prokaryotic stuff and you're having trouble, then you should reach out to Mrs. Smith or I. Jump into a Zoom session with us or whatever to help you through that. But if you tune to these little live stream today, tomorrow, Monday, and Tuesday, we will get through the unit before break, and they'll have nothing to do a break, which would be fantastic. All right. Plant cell and animal cell. I think if I were tasked with doing this, the first thing that I would about is there are some organelles that are only found in the plant cell, and there is one that's only found in the animal cell. And so I would look at those things first. And then if I look here, I see these two lines only pointing to things in the plant cell. And so I know the only things happening in the plant cell is Walker's here. She says something's happening with mics. One second, Emma. Thank you for saying that. Nice to see you. Hate to do the live stream, but I'm going to shut down a whole bunch of stuff.
Emma, is it just Mr. Suter or is it me as well? Both? Mostly Mr. Suter, because I hear myself echoing on Mr. Suter's end, not even on my end.
Okay. I'm shutting down some things. I didn't think. It's been a while since we did this.
Do you want to flip over to me for a second and I'll do the other diagram and then we can come back and see if that helps and you just mute yourself as you try to close stuff, or what do you want to do?
No, just 1 second. I mean it's almost
okay.
Mostly Mr. Suter. Oh man. Yeah, because I don't have anything else open. Am I still glitching?
No, it's not as bad.
All right, so Emma, am I still glitching out there?
Yeah, it's still glitching. You don't have YouTube open, do you, Suter?
No. I mean, I don't have anything open at this point. At this point, everything is shut.
Yeah, you're like repeating everything. Like that's what's happening.
If I get out, do you stay in? Do you remember?
I think so. Want me to try?
No, I'll have to hit end broadcast to get out. I was in reboot. Well, why don't you, if watching that bad, as much as I like to be the one to talk about, why don't you go through it quick?
Okay.
That's better.
Well, how about you leave that up there? I'll talk and you write. And we'll do it that way. Okay. So Mr. Suter was saying that he would start with the items that we know are only in one type of cell. So when you're looking at this diagram, I see on the plant cell at the top, there are two things that are labeled that are only in that plant cell. So I think about, okay, what is in the plant cell that is not in the animal cell? So first we, of course, have the cell wall, and then we have the chloroplast. So which one of those is the cell wall and which one of those is the chloroplast? Mr. Suter is writing it down right now, but that's okay. It's pointing to the cell wall. That makes sense. So cell wall and then chloroplast. And so some of my students were looking at this and saying, well, it looks like there's 3 lines there. Why are there 3 lines? It almost looks like there's 3 membranes, because then they looked at the next one down that goes to both of those, and it's pointing on the animal cell, and it looks like it's a double layer. So what is that? Well, when we talk about the cell membrane, we're going to talk about how that is a bilayer. And so it actually is 2 layers there, which is why it looks like that. I think that they're outlining it. So that one at the top there is going to be your cell membrane on this diagram. Now you can also see that as plasma membrane. So either term would work. The one that is only in the animal cell, the one that kind of looks like it's like fireworks, ta-da! Those are your centrioles. They're the ones that are really important and are going to be used in cell reproduction in an animal cell. They're not used in a plant cell because of that cell wall. So cell walls basically create another piece of a cell wall between the 2 cells, and then that'll just kind of like seal off. So they don't use centrioles in the same way. All right, so working our way down from the cell membrane then. The next one down is pointed to that area that's like basically everywhere in the cell. So like everything that is in the cell that is hanging out there is the cytoplasm. So the cytoplasm is essentially, I think about it like Jell-O. And Jell-O's mainly made of water, which is really important because if you think about water, you know, why would you not hop in a pool in the middle of a lightning storm? Well, because you could get electrocuted. It's a really good conductor. And so cytoplasm is mainly made of water. And that's really important because all these processes happen in the cytoplasm. And so we want it to be a good conductor. Think about it like electrical currents that are going to allow these reactions to happen. So we want it to be a lot of water in these cells. All right, moving down to the third one, it's pointed at these things that almost look like they're little holding spaces. Okay, that they're going to hold stuff, which is important because they are your vacuoles. Now, in an animal cell, it's just known as a vacuole, but in the plant cell, there's only one big one, and so we would call that the central vacuole. So that's why it looks so much bigger in the plant cell. Moving down to the next one that kind of looks like a little jelly bean, and it's really kind of hard to almost see what's inside of it. So I'm looking at the same type of shape in the bottom left-hand corner of the animal cell. It's a little bit easier to see that like kind of squiggle that's inside that is the secondary membrane of that. Okay, so I know that that picture that we're showing you is kind of dark, but that if you look down, it kind of has that squiggle inside. That's the second membrane of this organelle. And this one's really, really important for getting energy in the cell, and that's the mitochondria. And Mr. Studer's going to draw that nice portion there. And in bioenergetics, as you get to cellular respiration, we're going to talk about this one a lot. I'm going to talk about that cristae, that inner mitochondrial membrane. All right, moving down to the next one. These arrows tricked up a lot of my students, I know, because they were like, well, what's the first one pointing to? What's the second one pointing to? So I consider that first one to be pointing at the little tiny dots that are on top of that. And so those little tiny dots are the ribosomes. So the ribosomes are on part of the endoplasmic reticulum. So technically, that second arrow or that arrow underneath of ribosomes is pointing at the rough endoplasmic reticulum because it has ribosomes, where if you look kind of out from that, you have that smooth portion as well with no little bumps. That would be the smooth endoplasmic reticulum. But a lot of my students are just putting ER here or putting endoplasmic reticulum, and that is fine for me anyway. All right, the next couple of arrows all go together because they're looking at this central area of the cell where all this important information is happening. So a lot of my students didn't have a problem saying, well, that's the nucleus, but they were like, well, I don't know what each of those things are pointing to. So that first arrow is kind of pointing to all that squiggly stuff that's on the side. So that's actually your genetic information, your chromosomes, your DNA. So I know I accepted DNA, I know I accepted chromosomes, I know I accepted chromatin, and I know I accepted genetic information. So basically, do you understand at this point that that's where all of our DNA or genetic information is? The next one down is looking at the outside membrane of the nucleus. And so that is the nuclear membrane is what I put for that one. And I'm sorry, Mr. Studer, if that's not what you thought it was pointing to, because I know that this diagram is a little bit tricky in there. So nuclear membrane. The next one down, that's the one that I labeled as just like, hey, this is the whole thing is the nucleus. So that entire portion I just considered to be the nucleus. And then the last one here, it's kind of pointing to that dark spot, which I feel like it's a shame Mr. Studer can't talk about this one because he loves talking about the nucleolus and how it doesn't really exist. But we point to this and we say, hey, this is the nucleolus. There you go. Nucleolus that is there. And so although it is a darker spot when you look under the nucleus, I would definitely talk to Mr. Suter about this and whether or not the nucleolus really exists and what kind of happens at that quote unquote spot. All right, moving down to the next one down there. It's pointing at these little things that are in the cell that kind of get rid of and clean up stuff. And so those are our lysosomes. And so if we talk about, go back to our chemical basis of life, we talked about processes like hydrolysis. And I know I talked to my class about lysis, meaning to kind of break. So a lysosome breaks up and gets rid of all of the bad stuff that's in the cell, anything that's dead or things that need to be cleaned up and kind of gotten rid of. And then lastly, the one that kind of looks like a Wi-Fi symbol there, that is the Golgi apparatus or the Golgi bodies. And so you can either go with Golgi bodies, you can go with Golgi apparatus. And so you'll notice that that's kind of close to the ribosomes and to the endoplasmic reticulum. And that is because that is going to take the proteins that are produced at the ribosomes, and then it's going to go out to the Golgi apparatus. Anything to add, Mr. Suter?
Yeah, so I did switch my mic to my earphones, but I doubt that will make it better.
It's better. It's a lot better.
Interesting. So the one thing that I would add, guys, ladies and gentlemen, is one of the things that I found a lot of students getting confused about was the difference between that and that. And so with any of this stuff, and we'll see this when we go over the next worksheet here in a second, if I draw a smiley face, we talked about this at some point maybe, but if I draw a smiley face, you can tell me well, if you can see it, you can tell me very quickly that where the eyes and the mouth are. And so all of these pictures, the things are kind of drawn the same. I guess what I'm trying to say is if you get an understanding of what you're looking at, it makes it easier to decipher what it is and why it works. And so just pointing out really quickly, like when we drew that mitochondria over here, if we were to draw the chloroplast, on the inside of a chloroplast, let's do it in green because I can. On the inside of a chloroplast, and we'll learn more about this, we have these stacks of things. And so what's actually going on the inside of a chloroplast is different than what's going on the inside of a mitochondria. And they become very easy. It doesn't matter what artist draws them. They become very easy to tell the difference. So if I switch my camera back, and I think, do you have that one that's printed out on your side?
Yep. I'm going to share my screen.
All right, so
are you flipping over to me?
If we add that to the stream, make that nice and wrong, but hi. Yeah, I got to figure out how to do this. There we go, and make that nice and big. You should be able to spot those mitochondria and chloroplasts really quick because they're the kind of big kidney bean shaped things, right? And then you just need to look at the inside of them and you can tell which is which. And then the other big giveaway is the fact that only one of them is in the plant cell. I'm sorry, only one of them is in the animal cell, and the plant cell has both of them. So it's really easy if you have both a plant and animal cell to look at to figure out which one's a mitochondria and then go over and figure out which one's the chloroplast.
So that's where I would start with this, is looking at those organelles there. And so this one is in both cell A and cell B. And so when I look at this, cell A, look at the shape, only has one single layer over here. It's got two things. So I'm going to say, okay, well, cell A then must be an animal cell and cell B must be a plant cell. And so then I know in a plant, when I'm looking at these guys that are only in the plants, then down here, this must be our chloroplast, okay, which means that these ones that are in both, that's going to be our mitochondria. And so I'm going to kind of start there, like Mr. Suter said. And then moving down, I'm going to look at the things that are only in each one of those again. And so I know that this looked like a little firework in the last one, but if it's only in the animal cell, then this would make sense that this is the centrioles.
And then another thing just to add to that is that they're typically you'll see two of them drawn side by side.
Yes.
So those little tips that like things that can help you out.
And then over here, this outer layer of the plant cell, well, this is going to be your cell wall.
And so, like, thinking about this, ladies and gentlemen, think about where things are and what things do as you start to label. And then in this worksheet, you're looking more at what things do. So Mrs. Smith just labeled the cell wall. Okay. So animal cell doesn't have one. What's on the outside of the cell wall? And where is that? Where would you go next, Mrs. Smith? What's the next most obvious thing in a cell?
I usually go personally for this really big central vacuole, just because it's so large in the plant cell, it kind of draws your eye there right away. Like I tend to start with the bigger things first. And so just to make sure, oh, so these little things in the animal cell on this drawing, they must be the vacuoles. I had some people that labeled those as the lysosomes this time on this one, but it's also, like I said, connected to this one. So this kind of tells me right away. The next thing that I kind of look at is I'm going to be drawn to the nucleus. And so then I'm looking for anything that's connected to the nucleus. So I've got this spot in the middle, which could be the nucleolus.
In this paper, we're not going to talk about why, but I'm going to tell you right now, this paper's wrong. Right? Like you couldn't have it drawn the way it's drawn on the paper. But we're just going to ignore that for now. We'll come back to it when you guys get smarter in another unit. And for now, I shall label that other thing on the inside, the chromosome, or the chromatid, or the DNA. Whatever you label it right now is fine. We'll distinguish between those things later.
Down here, I don't really like this because this is specifically pointing to the nuclear membrane. So I feel I have to write that. But really, all of these parts together are part of, like this entire thing is also the nucleus. So I would have taken nucleus here too. Just saying. All right, then I'm just going to fill in, you know what I mean, what's left. So I'm going to look at what this is pointing to. And so this is pointing to the endoplasmic reticulum again, or the ER. What's my next blank? All right, I'm going to look up here. This is pointing to that jelly-like substance. So again, that is the cytoplasm, or just because they could put that, they could also put cytosol, just to say, which would be cyto. So that tells you cell, and then sol, S-O-L. And then I'm going to come down here to this blank. Here's my kind of Wi-Fi type symbol again. And so this is our Golgi body, or our Golgi apparatus.
How did teachers teach that before Wi-Fi?
I don't know. And then how did teachers teach before Wi-Fi? And then this one kind of tricked up some people, but it's pointing to these little dots. And some of my students said, but wait a second, like their little dots are here. And so this kind of makes a good discussion. Yes, there are a lot of ribosomes that are on the rough endoplasmic reticulum, but there's also these what we call free ribosomes that are hanging out inside of the cytoplasm. And if you take AP with me, I'll kind of explain why. So certain proteins are made on these ribosomes and certain proteins are made here. But that's not something at this level that you need to know. You just need to know that these are pointing to your ribosomes.
And so this is this really, if we really wanted to get into it, we'd start talking about where they're made, because you know where they're made. You put down that they're made in the nucleolus, and we would get into that. And then I could get into that thing Mrs. Smith knows I'm dying to get into, but I'm not going to.
All right. So hopefully you're still hanging with us as we go through. Okay, don't give up on us yet. So this next part, it says use red for the part that gives the cell shape and holds the cytoplasm. And there's also a line here. So yes, you want to color it, but you also want to think about what this is. So what gives the cell shape? What holds the cytoplasm? And so when I think about this, I think about cytoplasm as being mostly water. And what gives water its shape? So what, you know what I mean? If I put water inside of a glass, then water is the shape of the glass. If I put water into a baggie, then water is the shape of a baggie. And so it's about what's holding the water. And I'm thinking, Mr. Studer, that if we had our students last year, right now, there would be at least 5 of them that would have posed the question, is water wet? I'm just saying. Anyway.
Well, not only that, but a whole bunch of them would have responded in the comments with what the correct answer is. And that's killing me that we can't ask. Unless Emma's still here, and I know Emma would know the answer to that.
So what gives the cell shape and holds the cytoplasm is going to be our cell membrane. So then I would grab my red and I would go up here to my cell membrane, which is on the inside for the plant cell. Okay, it's the inside membrane, and I'm going to just quickly color. And then over here, I would label.
And while she's doing that, if you're at home, we understand you might not have color pencils, especially if you're doing it online. So we'll be all right if you don't color this diagram.
I'm just showing you because a lot of my students didn't color, and so I wanted to give them the opportunity to see what it would look like colored. All right, the second question says, use green for parts that make food. So what this is talking about is, do animals make their own food? Nope. So it's got to be our plant cells. So we're talking about a part that's only in plant cells, and some of you might be through this part, but I guess you're probably not watching this if you are. But we're talking about the process that is photosynthesis that happens inside of our chloroplasts. And so quickly, doing like quick coloring here, we're going to color those green. And yes, it's bothering me that it's not perfect, but that's okay. All right, for the next one, it says use brown for the thick outer covering that protects and supports the cell. So in the animal cell, it already has a cell membrane. We already said that holds the shape. So the thick part that's going to support the cell, this is why plants don't just fall down. Okay, that's going to be our cell wall. And so you're going to take brown, and you would be coloring in the
Coloring with Mrs. Smith.
Coloring with Mrs. Smith. You want to color more? Take anatomy. Color lot in there. The next one, use blue for the part that stores substances. So that's about what's going to hold. So like what on here is the biggest part that's going to hold? Okay, we're talking about the vacuoles right there. Okay, the vacuoles. So I'm going to take my blue. Shade that in, and then I got all these little blue vacuoles over here. Okay, next one. Use black for parts that release energy from food. So the releasing energy, that's my key one right there. The mighty mitochondria. Okay, and as I'm coloring, we can talk about the cells that need more energy are going to have more mitochondria. So we'll talk about that more in bioenergetics, but I know I talk about how your muscle cells need a lot more mitochondria because they need a lot more energy.
Always a question on the keystone.
Always. Okay, use purple for parts that carry hereditary information. So this is your genetic information. So these are your chromosomes. Your chromosomes, or chromatin or DNA, if you put that there.
You just wanted the color.
I did. I color all the time with my kids. It's fun. Next, use pink for a cell part that helps with cell reproduction. So if you remember, I kind of said this earlier, that cell reproduction in the animal cell is going to be the centrioles. Okay, that kind of looks red on the camera, but it's pink. And the last coloring portion is use orange for the parts that package. That's the key word that I'm looking for, in store chemicals. So I try to kind of like underline the parts that I thought were important in here. So to kind of help you. So packaging is going to be the Golgi bodies or Golgi apparatus. And I'm going to put apparatus here because I put bodies everywhere else. And I need orange for that.
You put bodies places? Just wondering.
You know, well, at school I've got what, 3 skeletons in my closet, so guess so. One of them's real. All right. Man, it's been a long time since we did this. All right, the next one, list 2 cell parts found only in the plant cell. Well, that's easy. Now I have to go back up, and like we've already told you that's the plant cell. So now which are the ones that are only connected to this side? All right, so the cell wall and the chloroplast. And then last but certainly not least, where in a cell do most chemical reactions take place? If you're paying attention earlier, I told you this. It is the thing that is mainly made of water. It's going to be a good conductor. So it's going to allow all those processes to take place quickly. And that is our cytoplasm. Or, like I said, sometimes they'll trick you up and put cytosol. It's the same thing with cell membrane. They can sometimes put plasma membrane. Just trying to give a little help to anybody that maybe is here that's already done all this. Review for the keystone. All right, Mr. Suter, any other things about this one?
No, I think that's it. And oh, now I hear me echoing. I don't understand. Anyway.
Is anybody still here? Is Emma still here?
We got 3 people watching according to this thing, which that's normally wrong. But it's disappointing. But here's the thing. We were so excited to start doing this for you again that we decided that anybody that joined us and watched this video, if you went to Moodle right now and clicked on those 2 assignments and hit submit and typed, I was in the live stream, that we would give you points.
Comments, I'm here.
Right, that we would give you points for watching the video with us and doing the live stream with us. If you're watching,
do all this.
Yeah, if you're watching the recording of this, it will walk you through. You may do it on paper, take pictures, whatever. Post link to DocHub, post a picture, whatever, in Moodle, and we will grade those for you. So if you go in the next 5 minutes and write that you were here in Moodle, you get the grade without posting anything. If you're watching it later, you got to post it. And we're going to do one of these tomorrow. So maybe people will find out about that. Maybe people won't find out about that. But, if people show up tomorrow and do the assignment with us, we might be able to kind of push that assignment through. Because we would really, really, really like you to be done with cells before break for 2 reasons. One, we want to get your grades up. One, we want to get you to passing. We want you to be successful. And we also want you to have a holiday break where you don't have any biology hanging over your head, because it's so nice to go into break knowing that you're done with everything. So I think that's it. I think that's it for us. Little longer. We're at 34 minutes. We had that little glitch in the beginning. A little longer than we normally like to spend on this. Maybe tomorrow will be shorter. I don't know. We'll see. Hopefully we see you. It's a little depressing. Because between us, Lisa, what do you think? We have I think I have about 90 students.
Right. We should have it. Let's make it under what it normally should be. So even if we have 150 students, which is less than we have, but even if we said 150 students and we don't have anybody responding to us.
Well, Have a great day, everybody.
That's the way the cookie crumbles.